And they started off strong with the quiz. So, we ended up talking about germ theory of disease. Okay, this is Koch, right? And what he did was he was able to link that a specific germ or organism causes disease, right? And he did this with Vibrio cholera causes cholera, Microbacterium tuberculosis causes tuberculosis, Bacillus anthracis causes anthrax. So he did this with 3 different things, okay? Again, something like this would be difficult to do today, right? Due to ethical reasons, right? And what you would do, really quick for review, is you have to get patient 0, isolate different microorganisms, inject each one individually into a healthy organism. That healthy organism must become sick because of that isolated specimen, okay? Symptoms must match patient 0, and then once you isolate the sick patient, the culture should match the original patient 0's culture. And that's how we know this bacteria causes this disease. Okay. Some other contributions that Koch did to microbiology. He taught the first microbiology course in the University of Berlin. Okay. And he developed a way to grow bacteria in pure cultures. Okay. And then, okay, so going back to these questions, right? Is spontaneous generation of microbial life possible? No, right? It's biogenesis. Life comes from life. What causes fermentation? What makes wine taste good? Yeast, right? Yeast makes the good tasting wine, which is alcohol, and then bacteria is gonna make that lactic acid, so that nasty tasting wine. So how many of you guys anyone like wine? Wine drinkers? So you open a bottle of wine, you use that wine, and then you put it back in the refrigerator, and you come back to it a week later, and you try to drink that same wine. Is it still good? No, it's trash, right? Because you've already introduced bacteria to that wine. You store it. That's why it's nasty a week later. Okay, I found that out the hard way. Okay. And then what causes disease? Living organisms can cause disease. A lot of clicks in here. Okay. How can we prevent infection and disease? We have these guys, right? We already talked about Joseph Lister, right? And he used carbonic acid to sterilize his instruments and sterilize bandages in order to prevent infection. And then we have this guy, Ignaz Semmelweis, okay? This guy, story time, okay? Sad. So this guy was a Hungarian physician, and he was noticing that women under his care delivering children would die at higher rates than women who were having their children at home, okay? So, and when they were having their children at home, it was midwives, right? And so this guy, like, he's a doctor, right? He's like, I went to medical school. Why do my patients have a lower survival rate than those delivering at home? Like, that did not sit right with him, okay? So what he did was he started to shadow midwives, and he started to notice certain techniques that they would do. So with midwives, they would pass their ideas from person to person. So they would say things like, grab clean clothes, right? Wash your hands, right? These little things that were not practiced in medical school, right? People would go from working on a cadaver and going straight to deliver a baby, right? There was no hand washing. involved in that whatsoever, okay? So when he paid attention to that, he went back to his medical students and he said, look, you need to wash your hands after you work with each patient, okay? And what did they do? They laughed at him. They said, you are ridiculous, I am not washing my hands, that's preposterous. And basically everybody drove him real crazy, okay? What were these women dying of? They were dying of pleural fever, okay? And nobody believed in him that you should wash your hands, okay? He ultimately went insane, okay? Because maybe he had like some mental illness in his family, but he did go insane. He ended up in a mental institution, okay? Where he then died of pleural fever. Yes, that same disease that he was trying to fight so hard against ended him, okay? So that's super sad, right? If I could go to heaven and give anyone a hug, it would be this guy, okay? Why was his hand washing so controversial compared to Lister's sterilizing of the instruments? It's because this guy said wash instruments, this one you're telling somebody wash your hands, you're filthy. Didn't have a little bit more of a negative connotation, a little bit more of a pushback, right? So they did laugh at this guy, they did not believe him, but thank you. We now know, yes, you should wash your hands, okay? Right, when you go to a doctor's office, as soon as they walk in, what do? Sanitize. As they're walking out, sanitize, okay? So poor guy, think hand washing, okay? Florence Nightingale, she's credited for the field of nursing. So she kept a journal log of all of these techniques too that she would write, okay? Use clean bandages, use this, use that, make sure you do these, all these little techniques that were passed down. Jon Snow, no, it's not King of the North, okay? I'm still waiting for more seasons of House of Dragon. But this guy is credited for epidemiology. So tracking back the source of an outbreak, okay? Okay, so during this golden age of microbiology, we answered these 4 important questions, right? Good sanitation practices such as hand washing and sterilization will help prevent infection. Okay, a few other fields that emerged since microbiology, immunology, right? So the study of the immune system, which we'll talk about in a later chapter, okay? Virology, we'll talk all about viruses later as well. Chemotherapy, so typically when you think chemotherapy, you think of the use of drugs against cancer, but chemotherapy is just the use of any chemical to treat anything. And then genetics, right? We have a whole chapter on genetics. I believe that's chapter 8. Okay. Immunology, we give a lot of credit to Edward Jenner and Louis Pasteur. Okay, so Edward Jenner, this is a guy that totally unethical as well. What he did was he noticed that at this time people were dying of smallpox, right? And he noticed that milkmaids, right? These milkmaids, they would milk the cows. There was something called cowpox, and cowpox would typically infect the cows. So the milkmaids that would milk the cows, when they got sick with cowpox, okay, recovered, and then they got sick, were exposed to smallpox, they wouldn't get sick. So these milkmaids could care and tend to people who had smallpox. They're like, why is that? Okay, cowpox is similar to smallpox. So once the milkmaids were exposed to the cowpox, the body kind of banked that in that memory of the immune system, and so then when it saw smallpox, it was ready to attack, okay? We didn't know it to that detail at the time. We just know if you've had cowpox, then you can survive smallpox, okay? So what he did, so with this pox, you would get these little crusts on your skin, right? And so he got a healthy little boy, okay? He infected this little boy with cowpox, okay? He waited 2 weeks. Then he introduced smallpox to this child. Should we do that today? No, okay? But he waited 2 weeks, okay? Once he infected him with smallpox, did he survive? Yes, so this is where we came up with that inoculation technique, so, or variolation technique. So introduce a pathogen to someone in order to get this memory, okay? If he wouldn't have waited 2 weeks, and maybe he just waited 2 days, would the outcome still have been the same? No, we'll learn later you need 2 weeks. Your immune system needs 2 weeks to build antibodies, okay? But in his case, it was 2 weeks, right? I'm sure eventually they would have got it right, but luckily for this kid, it worked out, okay? And then pasture cholera, he's the one who found, he was working with chickens and cholera, and he found that the bacteria, when you left it out for a long time, it would get weak, right? And if you infected this weak cholera into a healthy chicken, and then you exposed it to like strong cholera, it wouldn't die. So he came up with the whole weakened form can help get immunity in that sense. Okay? But we'll talk all about this when we get into immunology. We have a full chapter just on it. We have 2 chapters on immunology. And then virology as well. We have a whole dedicated chapter. Viruses, this field didn't come to be until we developed the electron microscope, right? Viruses are nanometers, way smaller than bacteria. So if we can see them, how can we study them or know they exist, okay? And then chemotherapy, right? So just the use of a chemical to treat disease, right? It doesn't necessarily have to be cancer. We just always associate it with cancer, okay? Alexander Fleming, okay, he was a grad student and he was working with bacteria, okay? One day he looks at his plate and he notices there's this mold on his plate. And he noticed there was a clear zone between the mold and the bacteria, which we call the zone of inhibition. And when he got this plate, he was like, oh no, I ruined my sample. I have to toss this out, right? Garbage, okay? But when he noticed that zone of inhibition, he was like, wait, hold on. Why is my bacteria not growing here? What's going on here, okay? What he ended up growing was that penicillium fungus, right? What does this fungus make? It makes penicillin, right? That chemical that actually breaks down and prevents Staphylococcus aureus growth, okay? So he gets credit for discovering this antibiotic, okay? And then we rise into that field. Okay? Genetics, we have a whole chapter on genetics and on genomics too. So genetics is just the study on genes that are passed from one generation to the next. So genes that are inherited, okay? And then genomics. So genomics is the study of all of the genes in the genome and how they interact. Because we have about 3 billion base pairs, but 75% of them code for junk DNA, right? Junk DNA means it's not DNA that specifically codes for a protein, okay? But it's not junk, right? If it's there for a reason. So a lot of these other genes, they're involved in regulation, right? So promotion or inhibition of certain genes at one point or another. And we'll talk about this later as well. So just the difference between genetics versus genomics. And the future of microbiology. So one really cool thing is bacteriophages, right? So bacteriophages being used in contrast to or in replacement of antibiotics, because as we'll learn, when we talk about antibiotics in chapter 10, that, you know, it's an ongoing battle, and every time we introduce something, we get resistance. And so what's something else that we can try to explore, okay? Because there, you know, at some point in my life, or in somebody else's life, we'll probably get to that point, okay, where no antibiotics are gonna work. That's like my worst nightmare. Like being sick for something and then like no antibiotic works on what I got. Okay. Questions? Okay. So those last few slides are foreshadowing. Yes.
You kind of went through like a future after like the golden age of microbiology, and I saw lots of names like Watson and Crick that are like DNA and stuff like that. I know that like Pasteur and Van Leeuwenhoek and stuff, those are people that we have to remember, but like these recent ones that we went over right just now, is that something that we need to also have the same amount of emphasis on?
Once we get to those chapters, yes. So again, skip chapter 2, go ahead chapter 3. Okay, chapter 3 is all about cell structure, okay? So, we're gonna divide it into talking about eukaryotic cells first, and then we're gonna talk about prokaryotic cells, okay? There's not as many slides to eukaryotic cells because we're not that worried about that right now. We're worried about the prokaryotic ones, right? We're worried about the bacteria. So, compare and contrast, okay? What are some similarities between prokaryotes and eukaryotes, and what are some differences? Difference, right? Prokaryote has a nucleus. I'm sorry, does not have a nucleus. Eukaryote has a nucleus, right? What about cell wall? Do prokaryotes have a cell wall? Yes. Right. Eukaryotes? Depends. Some do, some don't, right? Plants, fungi, right? They can have a cell wall. Things that they have in common. They're both made up of DNA. Okay? The way that the DNA is double-stranded, right? But prokaryotes, since they don't have a nucleus, right? It's not enclosed in a membrane versus eukaryotes do. Eukaryotes have a lot more DNA than prokaryotes. Prokaryotes generally just have like one chromosome. Bacteria have one chromosome. Sometimes they have two, but most of the time it's one. Okay. So their DNA, no clear membrane, one single chromosome. It's circular and there's no histones that wrap around it. Versus eukaryotes, they have a lot more DNA and they're enclosed in histones, which are these proteins that kind of wind up, kind of like a yo-yo. So they wrap around, wrap around, wrap around. So you can fit a lot more DNA into that compacted area. No membrane enclosed organelles, right? So there's no Golgi apparatus, right? There's no Ruffy-R. Okay? Eukaryotes do, and we'll get into all of the organelles as well. For prokaryotes, if it's a bacteria, it has peptidoglycan as the cell wall. Eukaryotes depends, right? It depends. They for sure don't have peptidoglycan. If you're a plant, it's cellulose. If you're a fungi, it's chitin. If you're a protista, it depends, right? If you're an algae, cellulose. If you're a protozoan, no cell wall. Prokaryotes, they divide through binary fission, right? So they just grow, split in half, boom, 2 duplicate copies. Okay. With eukaryotes, we gotta do some mitosis. Okay. Some is sexual, some asexual. Okay. But it's not binary fission. Okay, who cares about the differences between prokaryotes and eukaryotes? Why do we care why they look different? Why we need to know how they're different?
With respect to what?
Why would, going into your field, why do you care how a prokaryote is built versus a eukaryote? Going into medicine. What? Yeah, so we want to know structural differences because when it comes to treatment, right, these guys kill us, okay? So we want to know the difference between a prokaryote and a eukaryote because a good treatment is going to kill these guys and not kill you, okay? So we want to learn those differences and then we want to exploit them, right? Because a good antibiotic is going to kill prokaryotes and leave your cells alone, right? We want to kill this bacteria and we want to keep your cells, okay? If you have a eukaryotic pathogen, such as like a fungi, it's harder to design a drug of treatment, okay? Because they are more similar. And when we get to our parasites, I'll talk more about that, but eukaryotes in general are harder to treat than a bacterial pathogen for that reason. Okay, so thinking of a cell, if you picture like an M&M, a colored M&M, okay? On the most outer layer, you have, you know, that little pink strip of your M&M is gonna have some sort of either a cell wall or some sort of sugar coating, right? We don't have a cell wall, so we have a sugar coat, or can have a sugar coat. Okay? If you get past that pink layer, what's next? It's gonna be right the shell, like the nut, right, the peanut, okay? That's gonna be the cell membrane, okay? And then once you get past that, and then it's the chocolate inside. Oh no, it's chocolate and then it's the peanut. I haven't had an M&M, right? It's a chocolate, and then there's more chocolate, and then there's a peanut in the middle. You know what I was thinking of? I was thinking of a Ferrero Rocher, because the chocolate is on the inside, right? Dang, I haven't had a candy. Okay. I need a candy. Okay, the most outer portion, that color is gonna be that cell wall. Then it's the cell membrane. Then inside you have that cytoplasm. So if we're thinking about some variations, in general, prokaryotes and eukaryotes, they have some similarities and then some differences. So they all, some can have a cell wall in eukaryotes, and then prokaryotes have a cell wall. Okay. Cell membrane, they all have. Okay. It goes cell wall, then cell membrane. Flagella. So some eukaryotes have flagella. Bacteria can have flagella. Okay. It just depends. They just work a little differently. Okay. So for example, algae, plants, and fungi, right? But protozoa, no. Animal cells, no cell wall. Okay. Plants, we said, is cellulose, right? Complex sugar. Algae is going to be cellulose as well. Algae also is made up of agar. So this agar sugar is what we use in the lab, right? Our media is agar. We get it from algae. Thank you, algae. Okay. Fungi is going to be chitin. It's kind of crunchy. Some have cellulose, but we're going to focus and say chitin. Okay, chitin. Some eukaryotes, right, such as animals and protozoans that don't have a cell wall, what they have instead is a glycocalyx. So a glycocalyx is a sugar coating. There's two variations to that sugar coating. It can be very organized, which would be something like a capsule. And then it could also be very disorganized, which would be a slime layer, okay? What does it do? It helps to protect the cell, so it serves as a layer of protection since it doesn't have the cell wall. Because that cell wall helps with like rigidity, right, maintaining structure, cell-to-cell attachment, and then cell-to-cell communication, okay? When you think of glycocalyx, think of like sticky and sugary, right? If I eat an M&M for dinner when I get home and I don't brush my teeth, and I wake up in the morning and I have sticky stuff on my teeth, that's that glycocalyx, okay? That's that sugary stuff that starts to build, okay? Eukaryotes, some have a flagellum, right? Protozoans can have flagellum. In human, the only one that comes to mind is like a sperm cell. Okay, sperm cell has a flagellum that helps with motion. Eukaryotes can have cilia. So cilia is kind of like a brush. So like your respiratory tract is lined with cilia, right? And that helps expel mucus. Okay. We also talked about protozoans that have cilia too, and that helps them move kind of like a beat-like motion. I have a video that I'll show you when we get there, but it helps move too. Prokaryotes can have flagellum. The structure of the flagella is different though. Okay? They're structurally different, but they do the same thing. Okay? Cilia though, do prokaryotes have cilia? No. No prokaryotes have cilia. Cilia is only in eukaryotes. Okay, so that's another distinguishing difference between prokaryotes and eukaryotes. Why do you need movement through flagella and cilia? To get somewhere, right? So, for example, when we talk about some of our parasites that reside in the water, protozoans, how do they move? They need to move, and then they go inside of you and start to attack you. It's super gross. Okay. Very common, like Giardia. Giardia has a flagellum. That's backpacker's diarrhea, right? If you go hiking and then you drink the water without cleaning it first, there's tons of cysts in there of different types of parasites.
These human neutrophils, taken from the blood of a graduate student, are mobile cells that will quickly migrate to sites of injury to help fight infection. They are attracted there by chemical signals that are released by other cells of the immune system or by invading microbes. In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source.
This is an immune cell, and so the
When the source of
immune
the
cell is
chemoattractant
going
is moved,
towards
the neutrophil
that
immediately
chemical.
sends out a new protrusion, and its cell body reorients towards the new location.
Which is important, right? When we get to like our immune system and we talk about how the cells have to migrate to that site of infection, right? You can also have chemo means chemical, taxis is movement, so movement towards a chemical. You can also have phototaxis, so like movement towards light. Your mucilary escalator, right? So they want to expel any mucus that can be trapped that you accidentally breathe in. Cilia, if you're a smoker, you could burn all the cilia you have, and that's where you have like a smoker's cough. It's because that cilia has just been burned through, been damaged. If you quit smoking, you can grow it back though. Okay. You also see this in pertussis. So pertussis attacks the cilia, and so that's why you have like this really bad cough as well. Important though, right? So we went through things that are above the cell membrane. Now moving towards the cell membrane. Okay. Cell membrane not the same as cell wall. Okay. Cell membrane is made up of a phospholipid bilayer. Right. We got our hydrophobic tails, hydrophilic heads. Right. They make a double membrane that helps for fluidity. Okay. We have components inside the cell membrane such as sterols, okay, and types of cholesterols. Okay. We have different carbohydrates and different sugars, and that helps for cell-to-cell recognition. So my cell can have a carbohydrate on it, and when my immune cell recognizes, let's say, a liver cell, it can see it and go, oh, that's a Stacy cell. Do not harm this cell. Or it can see a bacteria and go, oh, this is not Stacy. I'm gonna launch a response. Okay. So cell-to-cell recognition. Where's my title? Okay. Endo means inside. Cyto means cell. So how do cells bring things inside of the cell? Endocytosis. So here we have the cell membrane, okay? It can pinch around an area. Once it closes up, you made a vesicle. So you have a phospholipid within the cell, okay? That's gonna be a major way for transportation of substances inside the cell, okay? You can also have exocytosis. So when things exit the cell, okay? Phagocytosis is just a very specific term. So phagocytosis is when you do endocytosis, but you're an immune cell. Okay? So if an immune cell is doing it, it gets a fancy name, phagocytosis. Okay? There are immune cells that specialize in eating, and we call them phagocytes. So they're cells that eat. Okay. And they'll eat things like bacteria. Okay. They'll break them down with a lysosome, and then boom, trash. We keep some of that information to make a memory cell, and then the rest gets disposed. And then moving on from cell membrane, we have the cytoplasm. Okay. So cytoplasm is gonna create an area for work, right? We have things like our microtubules and our filaments that help support, give shape, and different things can be transported, okay, in vesicles throughout the cytoplasm. Nucleus, right? So getting into our organelles. The nucleus has a double membrane, okay? Two membranes around it. Has a nuclear envelope, right? There's these little pores inside there's little pores on the cell membrane that help for things to leave the nucleus, right? Maybe like a little RNA, right? When DNA is replicated inside of the nucleus, okay, you can make messenger RNA, and that messenger RNA is going to want to leave the nucleus, right? It's going to go on and be a template for protein synthesis. Ribosomes that are found on the rough ER, the ribosomes are what's actually making the protein. Okay? So rough ER has ribosomes. It looks kind of rough when you take the picture. Versus smooth ER, no ribosomes. Okay? So messenger RNA leaves the nucleus, gets on this ribosome turns it into a protein. Let me see this picture. This picture, looking at the detail and what it looks like, what type of microscope took this picture? Without looking at your notes, what do you think? it is electron, which, is it scanning or is it transmission?
Oh, transmission.
Transmission, right? Can see inside, split, not 3D. Okay. Yes, okay, so rough ER has the ribosomes, which make the proteins. You also have smooth ER that doesn't have the ribosomes. Smooth ER is more for making lipids and fats. Okay. Directly connected to the rough ER, we have the Golgi apparatus or Golgi complex or Golgi body. If you say any of those 3, that's fine. I wouldn't be that picky. Okay. What does the Golgi body do? The Golgi body, this is our like post office. So proteins can get modified in the rough ER, and then they can get shipped to the Golgi body through a vesicle. Okay. Once it arrives at the Golgi body, or Golgi apparatus, it's going to decide, it can further modify, and it can decide where it's going to get shipped, right? So a protein that's being made can get shipped outside of the cell, right? If you were making insulin, right, and that insulin needs to be released, you have the RNA leave the nucleus, goes on the ribosome, gets modified on the RIPR, then gets sent to the Golgi body, and the Golgi body will send it in a vesicle to leave the cell, right? So main function is modifying and shipping, okay? Shipping and transport. Think the post office. Again, can ship to another area of the cell, to the cell membrane, or any other compartment, or outside. Lysosomes. This is our garbage truck, okay? If something needs to be broken down into waste, lysosome is gonna do that, right? I mentioned earlier that we have cells that engulf the bacteria, okay? That bacteria is in one vesicle. That vesicle is gonna fuse with the lysosome, and now you have enzymes that degrade, and you have the bacteria in the same vesicle. So what's it gonna do? Chew it all up, okay? So lysosomes is gonna be our garbage. It's gonna break down anything you don't want. It can recycle, and it can, again, if it's an immune cell, it's for memory. In some cells, the lysosome is like the stomach, okay? So for some cells, the lysosome is how they get digestion, right? That's how they break down to get the nutrients that they need. Mitochondria. Who took this picture? It's TEM, transmission electron, right? Sliced in half, level of detail, okay? The mitochondria, for size reference, is about the same size as the bacteria cell, okay? Mitochondria also has double membrane, so 2 membranes, and it has its own ribosomes, and it has its own DNA. So it has mitochondrial DNA. The DNA inside a mitochondria is not the same DNA inside a nucleus, okay? And that's important when we talk about the endosymbiotic theory, okay? So just keep that in mind real quick, okay? Mitochondria own DNA, own ribosomes about the size of a bacteria, okay? And what does the mitochondria do? What is it known for? ATP, powerhouse of the cell, right? So it's really good at making energy. Okay? It does cellular respiration. Okay, so going back to the endosymbiotic theory. So the theory, okay, how we get, how do we have this mitochondria inside of a bigger cell? Okay, how is that possible? Why does it have these things? Okay, so the theory is, okay, there was a prokaryote, okay, there was a prokaryote long ago that was an arrow. It was really good at doing cellular respiration, and it got engulfed by another prokaryote. that was probably an anaerobe, okay? And not that efficient at doing cellular respiration, okay? And so what happened was this small prokaryote, when it went inside of the, got engulfed by the larger cell, started this little symbiosis relationship where now the larger cell now has a source for ATP, and then what the smaller prokaryote benefits is now it has a home, right, where it's safe from engulfment of another cell, okay? What do we have as far as information or evidence to prove this theory? The size, right? Why is the mitochondria the size of the bacteria? Why does it have its own DNA? And it has ribosomes, right? Ribosomes are not organelles. Bacteria have ribosomes. Okay? And this evolved.
Does it also have like the inner and outer mitochondrial layer?
Yeah, so it has a double membrane, right? So it had its own membrane, and then it gets engulfed. So it still it has two membranes once inside of the cell. Okay. This is the same theory for chloroplasts, right? Chloroplasts also roughly same size, also has double membrane. Right? So they hypothesize, or their theory is, billions of years ago, the mitochondria got engulfed, and that's how we have a cell that houses mitochondria in it, right? And then after that, at some point, engulfs a chloroplast. Okay, now you have a cell that has mitochondria and chloroplast. Okay, that's the endosymbiotic theory. So endo means eat. So one cell went inside the other cell. Okay. Now moving on to prokaryotes. Okay, that's everything you needed to learn about eukaryotes. So you have to know. I feel like this is a refresher. I do have a game. I need to find it. I do have a game to play where you can practice memorizing each organelle, where it's found, and then what it does. I'm gonna go find it and I'll post it. Okay, for fun. It'll just be a fun practice. Okay. When we move on to prokaryotes, okay, are we gonna have organelles? No, right? That's a distinguishing difference between the two. Okay. Do we have cilia? No cilia. Okay. So just really banking in similarities and differences. You can even do like a little Venn diagram to remind yourself the differences and similarities. Okay. I need a drink of water. Okay. Prokaryotes. Okay, prokaryotes. Specifically bacteria. Okay. The size of a bacteria cell. I already said a bacteria is way smaller than a eukaryotic cell, right? It's the size of a mitochondria. When we think about surface area to volume ratio, so a bacteria, its volume relative to area that it has to do work is actually really small. It's about 0.3. Okay, so it doesn't have a lot of space to do work compared to a eukaryote. So a eukaryote has a 3-to-1 ratio. So that means that it has a lot of space to do work compared to its volume. Why is that important? The more surface area you have, the more space you have available to do things like metabolism, right? Work, okay? Mainly like metabolism, okay? So if you think about surface area, think of all of the cell walls and all of the organelles. Those all contribute to surface area, okay? Eukaryotes have more, so they are more complex. They can do more complicated things compared to the bacteria. shape. Okay, finally, we're talking about shape, right? How do we categorize bacteria into different sizes, shapes, and arrangements? Okay. You can have, there's 3 different main types that we're gonna see in our lab. We have coxy. Okay, coxy is just a circle, a sphere. Okay, if you have a sphere, it's a coxy. Coxy can come in different arrangements. It can be diplocoxy, which means you have 2 circles together. 2 circles together. Okay. You can also have streptocoxy, so that means you have a chain of circles. You have tetrad. Tetrad is when you have 4. If you will be able to tell when you have a tetrad versus staphylo. Okay, staphylo is gonna be, it looks like a cluster of grapes. Okay, cluster of grapes is gonna be staphylo. Tetrad has 4 together. Strept is a chain, and then diplo is 2. Okay. So like Staphylococcus aureus looks like this, and it's purple. So you need to know the shape, and you need to know the arrangement for the first exam. Okay. Bacilli. Bacilli or rod. Okay. A lot of times in clinical settings, they just call these rods. They don't call them bacilli or bacillus. Bacillus is one, bacilli is many. A lot of times in the hospital, they just say rod. Okay. If you see rod, they're talking about bacillus. Okay. Bacillus. What?
Sorry.
Yes.
That's not what we saw under the microscope.
Some of you may have. Some of you had bacilli under there. Some of you had coxy. Okay. I saw a few different things. Bacilli comes in specific arrangements. You either have, yes.
So on an exam, can we say rod or it has to be just?
You can say rod or bacillus.
Okay.
I'll take either. Because I say rod a lot too. You can have single bacillus. Okay. You can have diplo bacilli, so 2 bacilli together. I will tell you though, none of our specimens that we deal with are diplo bacilli. If you see something like that, it's not. It is single, okay? What you're seeing is you caught the bacteria as it's dividing, okay? One more, I will still get this. Someone will write diplo, and it hurts me to mark it wrong. If you see diplo, it's single. That is my key for you on the exam, okay? Yes.
Why is that?
We're just not working with any,
Oh.
so I know it, so it's not. None of the bacilli that we work with is single. Okay. We do work with streptobacilli though, so you have a chain of rods. Okay. And then I wouldn't do this to you. We actually just don't have any in the lab. So if it doesn't quite look like, it looks kind of a little bit in between a circle and a rod, it's Cocobacillus. Okay. So a little in between the both. Okay. We don't work with any. So my rule of thumb is, if it's not a perfect circle, it's bacillus. Okay, that's my key. If it's not a perfect circle, bacillus. Okay. We also don't have single. Okay. There's also no single coxy. Okay, no single coxy. There's only diplocoxy. Okay. There's no staph bacilli. There's only staph coxy. Okay, okay?
So you said that there is single bacillus, right?
Yes.
But no single coxy.
No.
And then any chain greater than 3 or greater is strepto.
Strep, and it could be coxy or it can be bacillus or bacilli. Looks like grapes. 4 is tetrad.
And there's no
So,
try, there's no trial, right?
not for this, no. So, what you'll get, alright, once you learn how to gram stain, on the first exam you'll gram stain, and then you're gonna look in your microscope and I'm gonna say, what is the shape and what is the arrangement, okay? And what's the gram stain, right? Is it gram positive, is it gram negative? Is it cocci, is it bacilli? And what arrangement is it? Okay. Those are the 3 questions. And we'll get more practice, we'll get more practice. Arrangement is actually bonus points. So if you tell me the gram stain and you tell me the shape, that's full credit. If you can tell me the arrangement, it's plus 2 points.
So for the gram positive and gram negative, describe it as like what color it should be. Because isn't it depending on if it's more of a, then whatever it has like a really
Yes. So we'll talk all about how you distinguish between gram positive and a gram negative. But once we get there and we look at exam down the road, you'll tell me gram positive or gram negative. And we do base that off color, okay? I'll teach you guys about it, but just because we have shape right here, I want to let you know shape is important, okay, when we get there. So you'll tell me the positive, negative, and then you'll tell me the shape. You have that, boom, 10 points. Can you tell me arrangement? Extra credit, okay? And we'll have open lab too. So gram staining can be frustrating, you just have to be patient. Typically the Friday before your exam, so our exam is gonna be I think on a Thursday. So the Friday before, we do like an open lab where we have the micro lab open in the morning all the way until like noon, okay? And we take shifts, so it'll be, Tat usually does the morning, Milena does after that, and then I do the last shift. So we'll have the lab open, probably like 6 hours. No, 4 hours. We'll do 2, 2, and 2. So you can come in that day and practice gram staining if you're not comfortable with what we just do in class. Because we do a lot during class, but it's just extra. Like if you just like, I need to practice one more time, Stacey. I need one more time, you can come in that Friday. And you can also practice streaking too. So we'll have like open lab and you'll get to practice your streaking and you'll get to see your plate before the exam on that Tuesday. So if you work and that's something you want to do, I'm letting you know now, but you don't need to come to open lab to be successful either, because we do practice in class. It's just like, if you have like anxiety about it and you're like, I just need to practice one more time, you can come to that. And you're welcome to come to more than just my shift. So you can come during my shift, you can come during whatever time works for you. We just tell all our students, we'll arrange that at some point, but we always do that, especially for this one, because this one's very hands-on. It's the one exam that's really hands-on. After that, you're not really like doing anything.
So for the exam, they will always be hours that we have, or will there be
Yes.
some already under the?
You will have to gram stain yourself for the exam. I could ask other questions about pictures and quizzes and stuff like that, but you do have to be responsible for your own gram stain. So like if you don't gram stain it correctly, then does that mess up your answer? Yeah. So there's technique and there's some knowledge, okay? So just foreshadowing, but we will have practice, I promise, okay? Okay. We can have spiral, okay? So spiral, this is gonna be like syphilis, okay? Syphilis looks like this. We don't call it arrangement though. When we talk about spiral, we call it variations. So if I ever ask you what is the shape and variation, you know I'm not talking about cocci or rod. Right? Red flag. If I say variation, I'm already not talking about a cocci or rod, right? So you have vibro, right? Vibro, which looks like a little Cheeto, right? That's Vibrio cholera. You have spirillum. Okay, spirillum is a little bit longer and it's kind of curled, and when it moves like a corkscrew. Okay, and then you have spirochetes. Okay, this is our syphilis. This is also H. pylori. H. pylori is a spirochete. Okay. Spirochete, spirillum, again, somewhere in the middle, right? It's a little bit shorter. Spirochete is very long, very skinny. Think of like when you make a cup of noodle and you take out a noodle, that's spirochete, right? If it's shorter and fatter, spirillum, and then if it looks like a little Cheeto, Vibrio. Okay, those are the 3 different variations for something that's spiral. You're not gonna cram stain a spiral though. Okay. That's another thing. Okay. How do we feel about, before I move on to that? Shapes. Variation / arrangement. Yes.
So we're not gonna actively work on spiral arrangement,
You won't
But it might-
gram stain one.
right? But it'll still be on the exam.
Yes, yes. I'm just trying to throw out as many hints when you get to your gram stain station. That's usually when we get a little bit stressed out. And I'm like, okay, narrow down the options. What are you working with, right? Also, let's say you get the gram stain wrong, right? You think it's a gram positive instead of a gram negative. Could you still see the shape? Yeah, could you still tell me the arrangement? Okay, you didn't lose all points, right?
Is it possible for us to like deduct, if we get the staining wrong, deduct whether it's gram positive or gram negative depending on the arrangement and the shape?
So you wouldn't be able to tell, because you can, it's either pink or purple to decide. So it's a 50-50. Yeah. Can you have, for example, we work with gram positive single rods. We work with gram negative single rods. So you see a single rod, it doesn't necessarily mean that it's gonna be gram positive or it's gonna be a gram negative, because we work with both. Yeah. That's why it's also important that we get our microscope skills down, right? Because we need to focus in order to see that size and arrangement, okay? So you guys do have to focus the microscope on your own when you get to that question, okay? If you are struggling, be patient, walk away from that station, come back to that station, okay? Yes. It is timed in the sense that you have the whole class period to complete. I wouldn't personally spend more than 30 minutes on the gram stain. If you spend more than 30 minutes on it, then you're diving into other time. So I would clock those 30 minutes. If you're completely stuck, move on to other components of the test, and then save that for the end and come back to it.
We're not timed at each station.
You're not timed at each station, but you do need to be mindful because everybody needs to hit certain stations. But I'm not timing you at your station, which is different than AMP, right? AMP is timed at the station. That's how it goes. I mean, that's how it goes. I time my B11 students, so that's just how it goes. But this one, no, you guys gradually just move. So give yourself a little bit of sense. Okay, I'm ready to move on. You can always come back to a question when it's open. Okay, but I will say if you are there's 5 minutes left in the exam and you cannot focus your specimen, call me over. I will focus it for you at the cost of 2 points. How many points is this question total? 10. You can afford 2 points, right? It's better to get 8 points than get 0 points. So if you're completely dying and you're like, I cannot focus this, I cannot get it, I need an answer now, I'll do it for you, but 2 points.
This has to be something made of oil.
Yes, oil. I know. practice. Okay, that's why I'm letting you know now how good you have to be with it, so that when we're practicing in lab, we're really paying attention, okay, can I get it? And not just, can I get it the first time, but how are you gonna troubleshoot, right? If you get stuck, what are the steps that I tell you guys? Start from the beginning, right? If you're looking under your microscope and it looks all bubbly, too much oil, take it off, clean the thing, go back again, okay? When you see your specimen and it just cannot focus, just take your time, small, take your time. And then when you think you just can't see anymore, turn it one more time, okay? And it'll be there. I promise you it's there, right? And we'll gram stain tomorrow, okay? We're gonna start gram staining tomorrow, just so you already start dipping your toes in it. Don't get too crazy stressed out. It's just gonna be your first time exposed to it. And then from then on, I'll build in time for you to practice a little bit every day, right? Except for the day of presentations. That takes the whole time. Unless you guys go quick, then you could potentially grab stain that day. But yes, that's foreshadowing, okay? It's okay, it's okay. We will get practice, we'll get there. So questions on shapes? No, we're okay? Okay. Prokaryotic structures outside of the cell wall, right? We can have glycocalyx, same as the eukaryotes. A flagella, same as eukaryote, just built different. Axial filaments, these are important for when you're built like a spiral, okay? And then pili, which are important for attachment. And then fimbriae, which are for exchanging some sexual information, right? This is the only, it's not, I don't wanna say sexual because it's actually not blending of genes, but it's the only way that we're able to get some genetic variation. So genes, some bacteria, they can share a gene that they have. Okay, so prokaryotes can also have a glycocalyx. Glycocalyx, again, can be complex, very organized, and that's gonna be a capsule, okay? When your bacteria has something that looks like this, so this is a negative stain, so they stain the background one color, and then they stain the bacteria another color. And you see this circular white portion, that's the capsule, okay? The capsule is going, if a bacteria has a capsule, it makes it more virulent, makes it harder to kill, and overall more pathogens that have a capsule are more dangerous because of that, right? So if a bacteria has like a capsule, it's able to avoid phagocytosis. It's kind of like, let's say you have a jar of pickles and you're trying to pick up that pickle and it's slimy. Same idea. So it's harder for your phagocyte to engulf this bacteria. So typically more dangerous ones have a capsule. Okay? Not necessarily. Capsule, gram positives or gram negatives can both have a capsule. It's just this, it's usually thick and it's organized, so a nice circle. A slime layer is gonna be thin and it's gonna be irregular, okay? Think of like when you're hiking and you're going through a lake and the rock is really slimy, that's like a slime layer, right? Think of your teeth. When you have like that buildup, you forgot to brush your teeth, right? Not me though, right? And then you feel it, that sticky part, that's that slime layer, okay? Here we have a picture. This one, we have a streptococci, right? And it has a capsule, okay? Again, this is gonna help when you're identifying certain bacteria. So some of you, for your case study, you have a capsule, okay? Again, you're more virulent, right? More dangerous pathogens have a capsule, okay? One of you guys got a capsule in your unknown, okay? Again, what's that capsule gonna do? It protects it from phagocytosis. Ugh. Okay. And then this is just a process of doing a negative stain. Because if you just do a gram stain on something that has a capsule, which you'll do at some point, it's really hard to see that capsule. You can see it. I can identify it. But it's really hard to see without the negative back stain. It's very, very faint. When we get to that point, I'll show you guys, like, there is the capsule. It's hard to see, but it's there. In order to nicely see the capsule, you have to stain the background, right? A lot easier to see. Again, in class we'll do one, a gram stain, and I'll show you guys.
Okay, so for like a capsule, in order to like get the bacteria, do they have to like insert it to specific
You do, it's an added layer. So you do have to get through the capsule. That's why things that are more pathogenic typically have a capsule. They're harder to kill. You have to get through that layer. We'll get to that. We'll get to that eventually. When we get to antibiotics. Okay. But for now, no. More dangerous bacteria have capsule. Okay. And then same with the slime layer. So when you think of like a UTI, right? UTIs are super popular in hospitals, especially if you have a UTI. Bacteria loves to grow on plastic. So when you have a catheter inserted for a long period of time, you have this thick slime layer start to build, right? Also think of that as like plaque that builds on your teeth, right? If you wash it away really quickly, it's easy to get rid of, right? If you don't brush your teeth for a while and it starts to build up, you have to get through this thick slime layer to get to the main microbes. Again, who's gonna do this? It'll be antibiotics, and it'll be stronger antibiotics and a higher dose of them in order to break down that wall. So it is a factor of virulence for the bacteria, sucks for us. Okay. So sticky as well, but that's to encapsulate and then expel. These guys are gonna stick and they're gonna hold on for dear life, okay? And it's harder to kill them. Okay, so comparison, capsule, slime layer. Thicker, circular, usually thinner and shaped irregularly. It looks kind of weird. Not organized nicely. They're both glycocalyx. They're both a sugar layer. Okay. Just two forms of it. Okay, I'm gonna stop there. I was gonna do a 5-minute break in the middle and I forgot. So there's your 5-minute break at the end, okay? Okay, I'll see you guys tomorrow. Tomorrow's our Friday, right? For class. Yeah. Yes. It's just the way that they move is a little bit different. So like flagella and prokaryotes, they rotate. So they kind of propel, like it's a little propeller. And then with like a sperm cell, it moves like a snake. You do need energy, but they're built, they have a hook, and that hook is what allows it to move like a rotator. So it works differently than the We'll do these as a group, so your group will hold it down for you as far as these two. Cause you'll do plates as a group, and then when you come back, you're gonna come back and see the results of what we did this day. This day we're just inoculating, this day we're reviewing. So this is probably more important. Yeah. Yes, just to remind me, so I'm like, where is Zell? I'm like, okay, yeah. You're good.
Oh, yeah, it is.
Yeah.
Yeah, I think the second one is next.
You can guess now, like in general, the information. Yes.
Yeah, okay.
You want it?
Yeah.
Yeah, we think it's simple as.
Are you all together?
Yeah,
Yeah.
three.
Oh, okay. I don't have it memorized. Which piece that he said? Sorry, I'm back here.